Okay, so let's go ahead and take a look here. So Hershey Chase is where we left off, right? So this is basically, to kind of refresh the memory, we're basically trying to figure out what the inheritance molecule is, right? So at this point, Abraham, McLeod, McCarty kind of gave us a little bit of a hint. And then Hershey Chase, we needed a good slam dunk, right? So we needed something that we clearly could not sort of argue against, something that's a little bit more definitive. Last time we talked a little bit about how bacteriophage infect bacteria. And the important thing is bacteriophage are only made out of protein and DNA. That's it, right? So you can only have those two. Now, the important thing to understand this experiment in terms of what they're doing is to try to understand what they're trying to achieve. So here's what they knew. They knew that their phenotype, which in this case was infection, right? So basically the virus hijacks the cell. They knew that was inside the cell, right? Now, they also knew that that represented a different set of instructions. Those were viral instructions doing that. And so that was essentially the information molecule. So the idea was whatever the information molecule was, has to be inside the cell. The question is, what is it? We don't know if it's protein at this point, and we don't know if it's DNA. So we have to figure that out. And this is the experiment that they used in order to figure that out. So what they did, first of all, is they basically took an isotope of, I'm going to charge up my Apple Pencil here while I talk around it. They basically took an isotope of phosphorus, P32. It's a radioactive isotope. Now, it's not like uranium, you're not going to like light yourself up. It's actually fairly innocuous, but it's good enough to develop film, right? So if you've ever it's kind of getting harder and harder now to actually explain film these days, right? Because everything's digital. And so you don't actually develop film anymore. But if you kind of recall back in the days when you had like a dark room and you had to develop film, any kind of light exposure, any kind of actually energy exposure for that matter, would basically cause a black mark on the film, right? So it basically developed the film in a way that you didn't want to. So P32 had just enough energy to develop the film, just like light would. And not only that, but wherever P32 was, it would basically leave a black mark on the film. And so what they did was they basically took the bacteriophage DNA, so the DNA associated with the actual bacteriophage itself. And of course, that DNA has nucleotides in it, right? Nucleotides, part of it is phosphorus, so they have phosphate groups on there. And so what they did was they fed the bacteriophage radioactive phosphorus, or nucleotides that have a radioactive phosphorus on them instead of the regular phosphorus. And so what would happen is the bacteriophage would incorporate this into their DNA, and then they would essentially over time accumulate radioactive or glowing DNA, if you will. Okay, so that kind of highlighted the DNA component. Now they can do a similar sort of a thing with protein. So with protein, basically what they have is a radioactive isotope of sulfur. And if you recall, sulfur is the culprit that is in the sulfhydryl group of cysteines. Remember way back in the day when we talked about functional groups, that last functional group we talked about was sulfhydryl, was that SH group. And the reason why that was important is because that's the one that creates the little disulfide bridges in proteins. And so as it turns out, we can actually create a radioactive form of sulfur. In this case, it's an S35. And so just like the same way we did with the DNA, if we fed the bacteriophage radioactive cysteine, then it would incorporate the cysteine into its protein mix, and then that basically be sufficient to highlight the protein. And so obviously you would have some incubation times in here to kind of get that incorporated into the normal bacteriophage. But over time, you could create two populations, one population with labeled DNA on it, and one population of bacteriophage with labeled protein. Then what they did basically is they went ahead and they infected the cells, and then they tracked or they followed the molecule. Now that they can see the protein and the DNA, they can follow where it goes. The question is here is who goes inside, right? So whoever goes inside should be the inheritance molecule. So let's take a look and see what their experiments are. So on the top, you can see you have the S35 labeled bacteriophage. So here you can see on the far left, you can see that you have labeled protein. So all the protein pieces are the capsule, the sort of the lunar lander piece of it. So all this stuff here This is all protein that's been labeled and not the DNA. Notice it's kind of black and white inside. So they infect the bacteria, they let it inject whatever the inheritance molecule is, whether it's protein or DNA. So at this particular point, what they now know is at this particular point, your inheritance molecule is inside, right? Because that's what infection does. It puts the inheritance molecule inside. The question is, they don't know what that molecule is. So then what they do before the bacteria lyse, because remember before we said the bacteria eventually will reassemble viral particles and then it'll burst and it'll release a lot of other viral particles, which then goes and affects other cells. But before it gets the chance to do that, what they did was they basically blended it all up. It's a fairly inelegant process, right? So notice just a kind of Walmart brand blender there. There was nothing high tech associated with that one. You just blend the devil up. And what does that do then? Well, what that does is it basically beats the devil out of these guys here, right? So you kind of like chop all these guys up, but the cells remain intact. And then after that, you centrifuge it. Basically, means you essentially like a washing machine. You basically spin it. And what that does is all the heavy stuff goes to the bottom and all the lighter stuff stays at the top. Okay. Similar to what you do when you spin out your clothes during the rinse cycle. So what did they see then? Well, here's what they said. First of all, the cells are heavy. So the cells would naturally go to the bottom and form a pellet. And then the other stuff, that is to say the bacteriophage proteins, they've been chopped up. They're fairly light, and none of them bacteriophage are very small as well, so are viruses. So they're not going to be heavy enough to spin down to the bottom. So what they're going to do is they're going to basically stay up here in what's called the supernatant, and then the cells are going to pellet down here in the pellet. So what you've done now is you physically separated the outside versus the inside. What you see then is your label, in this case, when you label protein, is not inside the cell. It's outside the cell. How do we know that? Because it's in the supernatant. Okay. So that's pretty convincing, isn't it? Inheritance molecule has to be inside the cell. Proteins are not inside the cell. Therefore, proteins are not the inheritance molecule. Of course, then what do we say? We stop there, right? Because we're not fully trained scientists. And we say something like this. Oh, therefore, DNA is a genetic material. No, that's not what your experiment just proved if you stop there. What your experiment just proved if you stop there is not the DNA is genetic material, is that protein is not. That's what the first half of that experiment came up with. Notice, to extend your results at this point to DNA is the genetic material is actually an error in logic. It's a conclusion based on a premise that is not supporting it. It's called a non sequitur, right? Because that's not what your data is telling you. Your data is not telling you anything about DNA. All your data is telling you about is protein. By the way, very, very common mistake that's made in science. A lot. This is one of the reasons why we spend a lot of time in grad school, to have this beat out of us. And even then, sometimes we fall prone to this. Why? Because we're part of the DNA crowd, aren't we? We're like, this is my idea. I want it to be true so badly that even if I get a whisper that it's true, then I'm going to say, you know what? It's true. No, that's not what you just said, right? What you proved was that protein was not. That's not the same thing. What you have to do now is you have to do an actual discrete experiment that says that DNA is, right? It's called negative data. You can't prove a positive idea with negative data. And this is the reason why I mentioned things like this all semester long, like don't fall in love with your idea, remain objective, don't get so wrapped up that you make this particular error. It's a super, super easy error to make, right? Because obviously the strong suggestion is that DNA is the molecule, but that's not what you did, right? And this is kind of like where a lot of those pushback questions come from. You know, it's like where it's like with Avery McLeod McCarty. It's like, okay, so yeah, it's suggestive, but are you sure you got all that protein? Are you sure you got all that DNA? Right? Because you didn't actually prove anything. All you said was the stuff that you removed, not all the protein, the stuff that you removed was not the genetic material. It could still be protein, right? Because that's what your experiment didn't say anything about all protein. That's an assumption that you overlay on top of your results. It's a very difficult thing to sort of keep track of, but we try to hound dog ourselves often, all the time. And this is what happens. This is the first mistake you make when you fall in love with your idea. Also, to be honest with you, this is a difficult one to manage, like when most of your idea is modeling. And this is the reason why I always say caution as you start to reach back into history, like evolution, things like that. Caution. If you can't do a discrete experiment to positively prove your idea, then you need to sort of back off a little bit and treat it judiciously. It's for this exact reason. Okay, so then they had to follow that up with the actual positive, right? Which is in this case is a follow-up about DNA. Great. So you told us about protein. So now the protein's not, right? It doesn't necessarily mean the DNA is. So let's go ahead and take a look at this. So then they did reciprocal. So then they labeled the DNA, did this exact same thing, infected it, let the DNA, let the inheritance molecule get inside, blended it up, and then they spun it down. And what did they see? They saw the DNA inside the pellet. So what does that mean? Now you can say my experiments are consistent with DNA being the inheritance molecule. Because here's your testable predictions. If DNA is the inheritance molecule, notice, then, those are your predictions, I should see DNA inside the cell. Do we? We do. If, here's the other, it's kind of like a negative prediction. If protein is not the inheritance molecule, then, there goes your then statement, that's your prediction. I should see protein outside of the nucleus, right? That is the phage protein, and that's exactly what you see, right? Notice you have to do both. In sloppy science, and there's a lot of it out there, you do one of these experiments instead of both of them, and then you run to the presses and you say, hey, look at this, I just proved this, that, and the other thing. Then, of course, the presses, they get all excited about it. But then when you sort of simmer down and you go back and you actually do your full due diligence in your science and your colleagues do that, you realize that your idea is falling apart. All right. And then, of course, the presses, they don't publish that one, do they? You don't hear a lot about, oh yeah, guess what? That thing we published a couple of years ago, that turned out to be a bunch of hogwash. It turned out not to hold up. And that's the big problem, right? Because what you're doing is essentially you're lying to people. You're telling them that, we figured this out, but you really haven't. We've got the answer, but it's not the answer after all. And so that's my biggest problem with a lot of scientific discoveries. Okay, DNA structure. This is going to go quick, right? Because this is all review. Not going to spend a lot of time here. You guys should already have this in your database. That's up here, right between your temples. So DNA structure, we already know about this one, right? So first of all, we already know that DNA structure has a couple of parts to it, right? So we have the sugar component of it. So your 5-carbon sugar, in this case, it's a deoxyribose. You have your phosphate group, right? In this case, which is attached to that 5' carbon. You have your nitrogenous base, which is either adenine, thymine, cytosine, or guanine. So A, C, G, and T. And then, of course, the important thing that's associated with the sugar itself, so I'll kind of bring this one down here, is, of course, your OH on the 3' end of the nucleotide, right? And this is important. Why? Because this is your growing end. Remember, we talked about that in the macromolecules chapter. So here's what it looks like. So here you can see your growing end here, your 5' phosphate over here. And then of course at your 2', either you're going to have an OH there, which makes you an RNA, or an H there, which makes you a DNA. So this is deoxyribose, and this is ribose. And then of course you have your nitrogenous bases. This should all be kind of reviewish from the macromolecules chapter. Of course, one of the things we want to do is we want to take a little bit of a closer look at the bases themselves. So remember, we have 2 different types of bases. We have purines and pyrimidines. The purines, I always remember as the double ring structures because of like Purina, Purina dog chow. And for a while there, they were like making those like little Purina double dog dishes. And little dog dishes are kind of stuck to each other. The one's supposed to be the food bowl, and the other one's supposed to be the water bowl. Yeah, we like to pick up 1 bowl instead of 2. But it kind of looks like the little Purina dog dish, basically. And your purines are adenine and guanine. Of course, then you have your pyrimidines, which are single ring structures. You have things like cytosine and thymine and uracil. Notice that thymine and uracil gets swapped out in RNA, right? So that in DNA, your bases are ACGT, and in RNA, they're ACGU. So you swap out your thymine for your uracil. Now, the one thing that I always tell students, because you want to understand the relationship between purines and pyrimidines. So it's helpful to be able to keep the 2 of them straight, like which one's which, especially when you're taking a look at DNA in particular. And so in order to remember which ones are purines and which ones are pyrimidines, I remember my genetics professor at UC Davis gave us this mantra, which I thought was really dumb at the time. Pure silver taxi. That's an L. Wow, that's creative. Of course, I rolled my eyes, right? Being the young student that I was. But I'll be honest with you. The first image that popped into my head as soon as he said this was like a DeLorean, right? And of course, back in the day, DeLoreans were cool because they were the only stainless steel car. I mean, we weren't so far displaced from the Back to the Future movie series, right? So the DeLoreans were pretty cool. So I just kind of imagined like being at the airport and having this DeLorean pull up as a taxi, right? That'd be pretty cool. It'd beat the yellow cab, right? So you just get to jump into the DeLorean and they take you wherever you want. So I thought that was pretty cool. And that's the only reason, by the way, why this stuck was because that was a DeLorean thing. Of course, back in the day, that's nothing special now, right? Because everybody kind of does a knockoff of the DeLoreans. But back in the day, that DeLoreans were pretty unique. So now if you want to see an actual, like a real silver car, just go to like Dubai or something like that, right? I mean, they've got diamond encrusted cars over there. It's yeah, I know, right? Yeah, I've seen those YouTube videos. It's like, seriously, people, do you have anything better to do with yourself? I mean, honestly, it's just hysterical. Anyway, I guess, see, this is what happens when you have too much money and too much time. You become quite useless, right? So here's how I remembered it. Pure is for purine. R, what's the elemental symbol for silver? Ag, right? Adenines and guanines. Of course, who's the taxi? TC, which is the leftovers, obviously, and those are the pyrimidines. So that's basically how I remember the pure silver taxi. Now this becomes important because we're going to take a look at this here in just a little bit. So one of the things, of course, this is just kind of showing, this is a review, by the way, right? So here you can see you have 2 nucleotides coming together. And so you can see the formation of your 2 ester bonds that are essentially bonding this phosphate group together. All right, those are your 2 ester bonds. So that's, I don't want you to get too much into the organic chemistry of that. We've already been down that road. We've already looked at those ester bonds, so that's sufficient, right? The important thing is to understand is that on one side, you've got your 5' phosphate, and the other side, you've got your 3' OH. This is your growing end. So this is basically where you would add your next nucleotide down here. Why? Because that OH is what you need in order to do your dehydration synthesis, right? So that's going to be where that OH comes from. So ultimately what that means is as you grow, you're growing in this direction, in the 5' to 3' direction of growth. And that's because of that 3' OH. Now, let's take a look at some of the work. So we're at about 1952 with Hershey Chase, but in that same era, there were several individuals who were looking at DNA, right? And so one of the things that we knew about for sure, and scientists get really good at this, is they want to know, like, okay, what can I study to get me the Nobel Prize, right? That's kind of what they do. They're a little less grubby now. I mean, they just they're fascinated by things, so they just start studying it. It was true back then too, but this was one of those ones. Every now and then you'll have a topic where it's like, okay, this is clearly a slam dunk Nobel Prize. Whoever wins this race, whoever figures this one out, is going to be walking up and shaking hands, right? Figuring out the structure of DNA was one of those. Everybody knew it. Everybody knew it, right? So we were starting now, thanks to Avery McLeod McCarty in 44, Hershey Chase in 52. We were now starting to sort of wrap our heads around this idea of DNA structure. And so there's a lot of individuals working on this, sort of adding little bits and pieces to the story. It's kind of like a community puzzle, right? There's not any one person who solves the problem. It's basically the contribution of lots of different people. Some people will kind of put in a couple of pieces and that's moves you forward. And then somebody else will come in later and put in a few more pieces and that moves you forward. One of those peace offerings was, that sounds weird, right? One of those peace offerers was Chargaff. Chargaff basically was looking at the proportions of the nucleotides to each other. So what he found was the purines and the pyrimidines were equal. So not only that, but what he also found out was that the number of adenines was equal to the number of thymines. So for instance, if I had 50 adenines, how many thymines would I have? Yeah, he found that you had this relationship. Not only that, but he found that there was an equality between cytosines and guanines as well. So the number of cytosines equaled the number of guanines. Now, why is this important? This is important to the structure of DNA because what this basically suggests, and when you take a look at the structure of the nucleotides themselves, you can actually see this. What he was saying was that the adenines essentially and the thymines are associating with each other. Look at this. Look closely at these bases. You can see the elements that are necessary for a hydrogen bond, can't you? Because what do you need for a hydrogen bond? An electronegative atom. Usually in biology, it is oxygen or nitrogen, right? And then you need like a hydrogen on the other side. Take a look what you've got here. A nitrogen that can leave a positive hydrogen exposed. A nitrogen here, which can pick up negative charge. A nitrogen here, which can pick up negative charge. Take a look at guanine. You have a negative oxygen out there, right? That's a big one sticking out there. We know that one looks negative. Here you have a nitrogen, but it's going to make that hydrogen look a little on the positive side potentially. And you have a big old electronegative nitrogen that could potentially make those hydrogens look positive, right? So if you take a look at the pyrimidines, you can see same things true over here, right? So you could have these positive hydrogens out here, this negative oxygen, potentially this negative nitrogen. On thymine, you got negative here, and positive there. That's a big one, right? So you can see those sticking out there like a big sore thumb. Same thing with uracil, negative here, positive here, negative here. So why is this important? Because basically what this is sort of when you analyze this polarity, if you will, right, of these bonds and this charge, then what this creates is it essentially creates the foundation for hydrogen bond formation. Right? Just like the secondary structure in protein folding, right? Those negative oxygens and those positive hydrogens on the nitrogen allowed for that backbone to bend and interact with itself, because you're creating the foundation for those hydrogen bond interactions to occur. Same thing's happening here. When you create these sort of like polar sort of charged looking situations on oxygens and nitrogens, or the hydrogens associated with nitrogens, you're creating essentially the necessary stuff that you need in order to build hydrogen bonds. So what does that mean then in light of Chargaff's? Well, what that means in the light of Chargaff's rule is this idea of complementarity. So this is what Chargaff actually developed for us. Chargaff came up with complementarity of the bases. So what he noticed was that A's tend to associate with T's using 2 hydrogen bonds, and C's will associate with G's using 3 hydrogen bonds. Why? Because of what we just sketched out. This, by the way, would explain the reason why he sees the same number of A's as T's. Is because for every A in DNA, there is a T on the other side that's associated with it. So they complement with each other. Together, these are called a base pair. And if you recall, base pairs are actually what we follow in electrophoresis. So when we were looking at different size bands, we were looking at base pairs. The entire thing, right? Because DNA has 2, because it's a double strand. This was an important piece of the story. This is a big piece of the puzzle. Now, another piece of the puzzle that was before this is work that kind of dovetails with Rosalind Franklin's work. So what Rosalind Franklin basically discovered was that the DNA molecule is a helix. And so what she did was X-ray diffraction studies on DNA, which basically X-ray diffraction is simply you purify a molecule like DNA. The idea is you want just DNA, so it's got to be really pure. And then you basically shoot X-rays at it, and then you collect sort of the shadow that it leaves on the backside. Okay, so it's kind of like me trying to figure out who you are by seeing your silhouette. So it's kind of like that. And so the X-ray diffraction pattern that you see is actually down here in figure B. And it doesn't really look like much, but if you're looking at it, what this actually tells us is that DNA is some sort of a helix, but we weren't quite sure what that looks like. So what did Rosalind give to us? Well, Rosalind actually made some important discoveries. First of all, she noticed that the diameter of this helix is about 2 nanometers. So she was kind of like getting some of the vital stats about the size of this helix. So she also noticed that it makes a complete turn. So it's twist, right? So it's a twist. It makes a complete turn every 3.4 nanometers. So she kind of laid down some of the important parameters of it. But as it turns out, this little X-ray crystallogram turned out to be the key piece to the puzzle. So she'd been working on this, and let me just kind of introduce another character to this plot, right, to this chase for DNA structure. His name is Linus Pauling. And Linus Pauling was a brilliant guy. He actually, not at this particular point, but he eventually won the Nobel Prize twice. He got really close to winning it 3 times, which is unprecedented. The first time he won it for his discovery of alpha helix. So you may remember alpha helix was secondary structure of protein. So Linus Pauling worked on discerning the structure, that helical structure of alpha helix. That was his discovery. And he won the Nobel Prize for that. So you could consider him at this particular point sort of the expert, so to speak, on helical structure. So when Linus Pauling was looking at the data, what he realized is he was hot on the trail of DNA, as many were. And so he was getting close because he realized that DNA was a helical structure, but it was a little confusing, right? Because as we learned later, it was a double helix. But he was getting close because he knew what a helix looked like. He knew how a helix behaved. And so he was getting close, and he probably would have solved the structure had it not been for the outright theft and trampling of one Rosalind Franklin by these two bastards. Now, why do I give these guys grief? Because generally speaking, if you ask Joe Average, who discovered the structure of DNA? Now, assuming they don't say, huh? If they have any kind of an inkling, probably more than likely the names they're going to come up with are Watson and Crick. So Watson and Crick basically got all the notoriety, all of it pretty much, and all of the superstardom, as much as nerds can get. That was pre-Gates. And they got it all. And basically what they did, and the story is obviously hotly debated, but generally speaking, the reason why we see Rosalind Franklin's picture here and not Maurice Wilkins is because, as the story goes, the more aggressive versions of the story pretty much say that Watson and Crick pretty much moseyed on down the hallway and saw Rosalind's data and pretty much stole it, and from there were able to solve the double helix structure of DNA. So that was data theft. Now, some of the softer versions say, well, no, that's not true. It was a collaboration. You know, we shared the data and all that sort of thing. It doesn't really matter how they came up with the data. It's really the stinging part is not so much how they got a hold of this data as much as what happened after the fact. That's really, really what pissed off generations of scientists. Because what happened after this, for the most part, was your typical sexist, sort of male-centric, pissing on the woman thing. And so in the process of Watson and Crick getting all the notoriety, basically Rosalind Franklin got trashed and pretty much swept under the rug and essentially eliminated or attempted to eliminate from history. Watson and Crick went on to, yes, that is true, she was, but the only reason why she was recognized was because there was a generation of scientists that were pissed off. By the way, at the time, nobody talked about Rosalind. So she was pretty much marginalized and dumped on and pretty much extracted from the entire story. So not only did Watson and Crick not solve the structure of DNA with their own data, but the person that they got the data from, they essentially smeared into oblivion. That's what happened. By the way, there are 3 seats on the Nobel Prize. 3 people can share the Nobel Prize. That's the most. 2 of them were, guess who? Watson and Crick. Guess who the third one was? You would think the third one should be Rosalind Franklin, or maybe Linus Pauling, right? Because, I mean, he did a lot of work on helix, but who was it? Maurice Wilkins. So Roslyn was completely eliminated from conversation. This, by the way, and not, as it turned out, what was so galling was this was like such a super rock star moment. I mean, Watson and Crick went on to write books. I mean, they were writing, it's like, this is how we discovered it. Well, oh, did you mention how you stole data from Roslyn? Did you mention that part? Oh, no, yeah, you didn't, because you're a bunch of misogynist bastards, right? Who don't actually deserve your career. As it turned out, Watson turned out to be a real prick. Because he actually had, and one of our own professors has some interaction with this. I mean, she said he actually had to get fired. He got fired from some institutions because he was an outright racist. So Watson, at least, was a real scoundrel and a real piece of dirt. And they went on to get like the royal treatment. And nobody talked about Rosalind. Nobody. You know what? To make this worse, it wasn't just Rosalind. In the same era, the 50s, there was a brilliant woman geneticist by the name of Barbara McClintock, who was basically thinking her male colleagues into the ground. An absolute brilliant geneticist. The stories go, in my former PI at UC Davis, Scott Hawley, who's now in the Academy of Sciences, he actually worked with her in her older age, and he was a brilliant guy, and he was just blown away by Barbara's just intelligence. He had stories of like her just walking through a cornfield, just walking through it, and being able to tell you exactly what was genetically what was going on in every single plants of her corn. I mean, she was just a brilliant geneticist. She discovered a lot of great things, some amazing things actually that, you know, just blew our minds in genetics. And she was active in the 40s and the 50s. But nobody knows about her, do they? No. So the next generation that came along of researchers that came along, 70s and 80s, if you might notice in history, that's also about the same time that women's lib started coming around and equal rights for women and things like that was really hot. The ERA, the Equal Rights Amendment, was a big thing in the 70s here in the US. But generations of researchers after the fact were pissed off. I mean, literally pissed off, because we've been sitting there seeing what happened to Rosalind, and this was not okay. And a lot of us who were in genetics were seeing what was happening to like Barbara McClintock, and we were not okay. I mean, it was an angry generation, and it still is. And it wasn't until later, and you're right, I mean, she did get recognized later, but it was too late, because Rosalind had died, unfortunately, and they don't do posthumous awards for the Nobel. Luckily, they were able to award the Nobel Prize to Barbara McClintock, rightfully so, because Barbara McClintock was probably the most brilliant geneticist of that era. And if anybody was going to get the Nobel Prize for genetics, it should have been her, and she did finally get it, but she was like 83 years old. So finally, you know, they were gotten beat into submission, because, I mean, there was a lot of researchers who were just red hot angry, because a lot of us, I mean, we were in the labs of women researchers. And so it's like, we knew how brilliant these women scientists were because we were working with them. In some cases, we were them, right? You know, but we were, yes. Did you see the pictures I sent, Benson?
Yes.
Yeah, you be watched. See, you thought you were watching me. I was watching you.
Well, enjoy.
Yes, I know. The kids were like confused. They were like, that was really nice of them. I was like, guys, it's a joke. They never got the joke, but they were happy about, they thought that was nice. So it's like the toilet fairy came to visit them. So they were happy about that one.
Excellent.
Yeah, by the way, that's all being recorded.
Excellent.
So just saying that, just roll with it.
Can I just say hi to your class really quick?
Yeah.
Hi. Oh my God.
Well, the rest of them are online.
Oh, okay. Okay. I teach Bio 112. Oh, yes. So if you're interested, we're offering 112 online this summer, so it won't screw up your summer. You can actually do it all online. We have a lab kit that we send to you, and So if you're interested, Mark has my information, and my name is Fleur. I'm the chair of the bio department. So thank you. Okay.
Yep.
All right.
Okay, so that was a plug for bio too. If you were wondering, that's everybody online is gonna be like, what's going on? It's like, you had to be here. See, I'm just saying, there's benefits to being here. But you're right, that really like didn't stick well, because a lot of us were growing up in a college, we were learning in our college, and we were working with these women researchers, and we were just like amazed at like how brilliant they were and are and still, right? And so it just was, and we still something that just really gets at us. We're doing better now. There have been many more female Nobel Prize winners since then, but there's still a long ways to go. So, but notice, and the reason why I like to mention this is, and it's really galling too. I mean, it would have been irritating if these guys here, if these guys hadn't basically just sort of catapulted to rock star superstardom, then it would have been irritating, but we probably would have dealt with it. The fact that these guys basically just sort of almost immediately soared to like messianic heights, like almost like, oh my gosh, these guys are the ones to talk to about science. It's like they're the ones who are the tops. I mean, that's what was so galling, is that they basically took this and essentially did nothing useful with themselves. I mean, well, that's not true. Crick actually did make some contributions, to his credit. Watson wrote a bunch of books and pretty much got himself fired from just about everywhere it went. But this is what, the puzzle analogy, the community puzzle. Notice everybody's adding a piece to the puzzle, right? That means everybody has a contribution to the puzzle. But what these guys did was these guys came in and took the last piece of the puzzle away from Rosalind and then put it in there and said, like, look, I just solved this entire puzzle. How many of you guys would want to punch him? None of these guys would be conscious if I was in the same room with them. That's exactly what they did. And so I tend not to like them. So this is the reason why I always mention these sorts of things, right? I mean, we're people, we're humans. We live in a culture and a society. And so it's impossible to divorce science from society, right? Society affects science, science affects society. And sometimes not for good reasons. This is an example of society having a very negative effect on science, or really a very positive effect, if you think about it. Because if they hadn't been such bastards, and if we hadn't loved to hate them so much, then maybe we would have never gotten angry enough to right all the wrongs that women researchers were going through. And that to this day, women would be getting unrecognized for all the great work they're doing. So you could think of it one way or the other, right? Personally, I would rather Rosalind won the Nobel Prize with, but that didn't happen. Okay, so let's go ahead and take a look at the double helix. So in this particular case, now you notice here we have our strand of DNA. So we have our 5' phosphate over here on this side, and here's our 3' OH. And so our next nucleotide is going to be added here at this end. Now, a couple of things to notice, first of all, is notice you've got your phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar backbone. So there's your backbone right there. It's a common backbone. And you've got these bases sticking out. Right. The other thing to also notice is that in DNA, there's what we refer to as an anti-parallel orientation to it. What does that mean? Let's take a look at it. I'm going to write 1/2 of the DNA molecule. So there's my backbone. Here's my bases. I'm going to do A, C, G, T, A, C, G, T. How about that? So this is going to be my 5' end. This is my 3' end. What does the partner strand look like? Okay, so what goes with A? T, right? And you use 2 hydrogen bonds with that one, right? What goes with C? G. And there's 3 hydrogen bonds there. And G goes with C, right? So there's 3 hydrogen bonds there. T goes with A with 2 hydrogen bonds. A goes with T with 2 hydrogen bonds. Of course, there's another T there, right? With 2 hydrogen bonds. C goes with G with 3 hydrogen bonds. G goes with 3 hydrogen bonds. T goes with A with 3 hydrogen bonds. Now, what's the complement to 5 prime? Three. Notice the anti-parallel. One's going this direction, the other one's going in this direction. That's anti-parallel. So here's your DNA molecule. So now you can see Roslin figured out 2 nanometers here, a 3.4 nanometer total turn. And then of course you've got Chargaff's rule pairing up the 2 complements to each other in between to complete your double helix. And so to kind of get an up close of what Chargaff saw, here's how your complements work out. So your guanine has the negative snuggling up to the positive here of your cytosine. The positive hydrogen over here and the negative nitrogen over here is going to form an H bond. The positive hydrogen here and the negative oxygen here is going to form an H bond. So those are your 3 hydrogen bonds that you get between C and G. Between A and T, you've got that positive hydrogen over here and the negative oxygen. So that's going to form your first hydrogen bond. Of course, your negative nitrogen and your positive, because why this is negative here. So it's kind of sucking all the electrons over there. So it leaves this positive. That's going to make your second one. Now, a lot of times students be like, well, wait a minute. What about this one here and this one here? This is negative, right? Doesn't that snuggle up to something? Well, the fact of the matter is it doesn't. Number one is because that's only a carbon. So it's not as electronegative. And all of that, but this is too far away once you orient the other 2. Okay. So that's basically how your complementation works. So now that we got the nuts and bolts down, what about DNA replication itself, right? Let's take a look at the mechanistic underpinnings of DNA replication itself. So the first thing that we noticed in DNA replication is that there was 3 basic models of how this could happen, right? So here we have a DNA molecule, and we have this kind of existing DNA molecule. So the question is, how do we make a copy of it? Well, we have 3 different plans. So the conservative model basically means that you would essentially make the new strand from scratch. Right, so the old strand stays intact, and you make the new strand from like nothing. But there's a problem with this, right? Where does the information come from to make your new strand? Right. So that's a problem. I mean, because if you want to think about like, the evolution of molecules, right? I mean, the idea is it took forever for this first DNA strand to come together randomly to create a functional gene. You're asking essentially for that to happen again. I mean, in the absence of some sort of a plan or some sort of information, right? Instructions, if you will, in order to build this, if you don't have that, which presumably you don't because the original strands stay together. So they're not available, right? So in order for you to build another one requires you to essentially recapitulate evolution that took an awful long time the first time, will take an awful long time the second time, and the third time. And you're asking this to happen. How many times do we go through replication? Well, how many times have your cells divided by the time, since you were a single cell? A lot. So you're asking essentially evolution to recapitulate to itself every single round of mitosis. Is that going to happen? Of course not, right? Now then there's the semi-conservative model. So basically, in this case, you have one strand remains intact, but the other strand will basically act as template. So what happens is you generate two template strands. From the old DNA. Makes a lot of sense, right? Because that would solve your information problem, wouldn't it? And then they had the dispersive model. Basically, this is kind of like where you have sort of a patchwork. Of the old and new. Not quite sure how they envisioned this one working, honestly, because it just like seemed dead in the water to me. So of these three, if you reason it through, right, you would imagine that really there's only one that seems viable, right? Let's take a look at what we're talking about. Here's how the conservative works. So here's your old strand. So they're basically saying your old strand stays intact, and then you basically create a new strand from scratch. Well, that sounds great, but guess what? If you're going to do that, then generally speaking, what that means is you have to have some sort of a, not an intelligence, but some sort of a pre-existing plan, right? A design. The only reason why we do this is because we've got a brain that can basically say, I can make a duplicate copy of this, right? So it's kind of like that sort of recreation event, right? You actually have to have an intelligence in order to drive this, or information in order to drive this. And so that's the reason why they're looking at this. They're like, well, yes, probably isn't going to be happening. And then, of course, you have semi-conservative, in which case you have your old and your new mixed together, because the old is acting as the template for the new. And then, of course, finally, the dispersive basically is where you kind of have this patchwork of old and new sort of interspersed between, kind of like a quilt, where you have old and new DNA patched together. Not quite sure how they envision the mechanism of that happening exactly, but that's just what their model was initially. Now, if you take a look at this, what we're going to do is we're going to basically do an experiment. This is the Michelson-Stahl experiment, which was happening in 58. So in 1958, I mean, you're kind of heading toward the end of the Eisenhower, right? The Leave It to Beaver era, the Red Scare era in the US. Everybody was afraid of communists. And we were rounding up all the Hollywood people in the droves because we thought they were all communists. Of course, now we should probably round them up as well, mostly just because they're generally useless for society, especially if all of our movie theaters are closing. So why do we need them around, right? Just to laugh at mostly, and be glad we're not them. So basically what they did, by the way, just to let you guys know, at this particular point, this kind of, because a lot of people will know this name, not Eisenhower necessarily, or certainly not Adlai Stevenson. But at this particular point, there is a young man who just won the US Senate seat in Massachusetts by the name of John F. Kennedy. So he is 2 years away from running for president in 1960. And so a lot of us kind of use that Kennedy administration as kind of like our before and after. Like there is the world before and then the world after Kennedy, and the two were very different because we just kind of unraveled after Kennedy and we were all uptight before Kennedy. At the same time, the vice president was Richard Nixon, who would later go on to be the president 10 years later, who actually ran and lost against Kennedy in 60. But the reason why is he was the vice president. So at this particular point. So, by the way, if you're wondering, a good comparative for this, very similar to what happened with Clinton Gore. So you had Clinton, a relatively popular president with a very, well, a vice president who lost in 2000 to Bush, who was a more charismatic individual than Gore was, certainly by a lot. Maybe not as smart as Gore, but certainly more charismatic than Gore. Exact same thing happened. Exact same historical pattern. You had a popular president, Eisenhower, with the vice president, Nixon. And then the vice president ran in 60 and lost who? To a more charismatic individual in Kennedy. Okay, very similar patterns. If you want to understand what's happening, don't freak out. Don't read your prophecies. Just read history. We tend to just basically recycle the same thing we did. And we do the same thing over and over and over again. We are not smart animals. More lemming, really, than human. Let's take a look at the Messelson-Stahl experiment. So what they wanted to do is they wanted to tease out the difference between these three models, right? Conservative, semi-conservative, and dispersive. And so the way they did it is, and here's the challenge they had, right? The challenge they had is like, well, we could do this if we can figure out some way to differentiate between the old strand and the new strand, right? But how can we do that? So what they did is actually they used an isotope of nitrogen, right? So here's normal nitrogen. So N14 is normal nitrogen. If you look on the periodic table, nitrogen has a mass of 14. So that's normal nitrogen. N15 is heavy nitrogen, right? Because it's got an extra neutron. So it's going to be a little on the heavier side. So what they did was they basically grew bacteria in heavy nitrogen. So what does that mean? That means all the DNA that these bacteria were making was going to incorporate heavy nitrogen N15 into their bases, right? Their purines and their pyrimidines. They were going to incorporate heavy nitrogen N15 into those bases. They allowed them to do that for a little while. And then they switched them to the light nitrogen media. So now here you have bacteria that have been making their DNA with heavy nitrogen this entire time. And now you switch them to a light nitrogen source, the normal nitrogen. And then what you're going to do is you're going to analyze your results. So what did they do? So here's what they basically said. If this is N15 or heavy nitrogen, then that means when we switch you, here's your switch, then all of your old stuff should be heavy, yes? And all of your new stuff should be light, because now you're making it on N15. Well, guess what? If I run this out on a gel, after that first replication event, I should see a band that is heavy, and I should see a band that is light. Right? So this is after your first replication. This is what they're thinking. However, if you keep it on light nitrogen, right, this is your N15 or N14 treatment down here. If you keep it on light nitrogen and have it go through another round, right, then basically what you're saying is this guy's gonna, you're gonna make essentially another copy, right? So you're gonna make a new, and then a new old, right? Because this one doesn't ever change. So you would expect then over the second round for your light to get a little darker and your heavy to start disappearing, right? Now, that's what you would expect for the conservative. Notice what I'm doing. I'm trying to hypothesize. This is a type of hypothesizing. If this is true, then this is the pattern I should see. Now, let's hypothesize with the semi-conservative. So after I switch in the semi-conservative, what should I see? I should see not light, not heavy, but something that is in between. So here's where my heavy band would be. This is where my light band would be. What I expect with semi-conservative after the first round of replication is something in the middle, right? Because it's half and half. None of that. In the second replication, I would expect to see the same thing. Or is that true? Let's think it out for a second. If these guys were to split templates, one would be heavy, right? Old, blue. One would be new, old, new. Then this strand here would be a combination of old, new, and this would be new, new. This one here would be old, new, and then new, new. Notice what's happening. I've doubled up my new. So now I've got a little bit of light coming through, right? So this guy starts to get more pronounced as I go on, and this guy starts to fade as I replace it. So that's kind of what you would expect to see with semi-conservative. With dispersive, I have no idea what they expected to see with that. I mean, it's going to be something in the middle, but it depends really on how much old you have in there and how much new, right? So this could be anywhere. It could be a smear, actually. If you've got a lot of DNA molecules with various amounts of old and new, it wouldn't come across as a band. This could basically come across as a smear because you got every kind of potential size in there, but not a discrete size. Okay. So let's see what they got. So here you can see that basically you're going to see two different densities after the first round. Guess what? They didn't see that. So that's out the window. In the dispersive model, you would expect to see maybe an intermediate band in there, right? Because you got a little bit of each. But in the second band, it kind of broke down, right? Because what happens is you would expect to see a single band as it starts to make new, more newer. And so that's not what you saw. You saw actually two bands. Why? Because the two bands were the growing light band. That's the second band. Plus the disappearing medium band. So all their observations were consistent with semi-conservative. That's kind of what it looked like. So basically they took their bacteria, they grew them up in N15, and then they switched them to N14, and then they just allowed them to go through their replication. So, you know, every 20 minutes or so, they go through. The doubling time for E. coli is about 19 minutes or so. So this, you know, after the first round, after the first 20 minutes, they would harvest them, and then they would run the gel. And then after 40 minutes, they would harvest them, and then they'd run the gel. And so what they saw was, before replication, they saw the old heavy band, because that's all you had there. And then after the first round, they saw the medium-sized band, right? Because it was part old, part new. And then after the second round, they saw the light band starting to come out now, right? And the disappearance of that medium band. So this is the data that they saw here. So here you can see this is the heavy going to intermediate, going to the both of them. Okay. So let's take a look at the mechanism of replication. A couple of things you're going to need, right? So obviously, you need something to copy. That immediately eliminates the conservative model, right? So you need some sort of information. You need DNA in order to be able to copy it. Otherwise, it's not replication, is it? It's creation, right? That's the problem with the conservative model. It's not replication. It's creation. If you're creating something from scratch, that's creation, not replication. Replication means you're essentially making a copy of something that already exists. So that means you need a copy of something that already exists, in this case, the DNA molecule. You also need somebody to do the copying, right? So this is where you get your enzymes of replication. We'll talk a lot about those guys. And then you need the building blocks. These are your nucleotides. So you need all of those for replication, right? You need the information to copy. You need the mechanism or the machine to do the copying with, that's the enzymes, and you need the materials to copy it, right? The same thing basically happens with like a copier machine. If you think about it, I have something that I want to make a copy of, like your guys' quiz. I need some sort of a machine to do the copying, that's the enzymes, that's the copier itself. And I need the materials in order to do that, right? That's like the toner and the paper and stuff like that. I need all three of those together in order to get a copy. Right? If I'm missing any one of those, guess what? I don't have a copy. So those are all necessary requirements. Now, when you take a look at DNA replication, and pretty much everything from here on out, we're talking about DNA replication, we're talking about transcription and translation, which is going to be the next chapter. There is a common three-step mechanism that you want to burn in your brain. They all have it. They all start off with initiation. These are preparation steps. How do you prepare for the actual event? They all have an initiation. There's a replication initiation, there's a transcription initiation, there is a translation initiation, and there are events that happen in each one of those during initiation to get yourself set up. After you get initiation events set up, then there is elongation. Elongation is the actual execution of the process itself. In this case, there is initiation, that is getting yourself set up and ready for replication. And then there's elongation, which basically means you're actually putting together nucleotides to form DNA. That's the elongation. There's a transcription elongation, there is a translation elongation. Okay. And then of course, all good things must end. You have a termination event. So you have a replication termination, that is to say the point at which you stop because you've already completed or finished your replication. There's a termination for transcription. When you're done transcribing a copy of your DNA, and there's a termination point or an end to your translation, where you basically end your translation event. So all three of these major mechanisms, which is DNA replication, transcription, and translation, all have the same three-step mechanism: initiation, elongation, and then termination. Okay. Now, most everything we talk about then with respect to these three fits into one of these three major phases. Okay, so let's just kind of take a look at what the task is first of all before we start talking about the initiation events. So this is kind of what you're doing, right? So basically what we mean, to kind of follow up with the semi-conservative situation, what we basically mean when you're actually building the molecule is, notice you have your 5' phosphate here, your 3' OH here. But notice it's antipolar, right? So what's going to happen is you're going to only add to your 3' growing in. So what this means then is that in this particular case, since adenines are complementary to thymines, then you're going to bring in a thymine base pair. You're going to remove these two guys here. That's going to leave as what's called pyrophosphate. And then you're going to form your bond between that phosphate, that new phosphate, and your 3' OH. That basically locks in your new base pair. So then the next step is you read this next base pair and you say, oh, hey, guess what? Adenine, that goes along with a thymine. So you recruit another thymine in here and you basically add it to this growing in. So essentially what's going to happen is you're growing in this direction. And who's doing that? The first, and probably the most important, of the enzymes of replication, your DNA polymerase. In this case, there's 3 different DNA polymerases. This is what happens to be the work of DNA polymerase 3. And we'll talk about the functions of DNA polymerase 3 verses 1 and 2 in a second. So let's take a look at this. How do we initiate? Notice the term initiation. So these are initiation events. How do we initiate synthesis or DNA replication? Well, first of all, it's important to always understand that number one is that you're only going to be adding nucleotides to the 3' end, right? So that's your 3' growing in. And that means you can only grow in the 5' to 3' direction. So this is basically a kind of a polarity for your polymerase. It only has a forward direction, 5' to 3' direction. There's not a reverse direction. It can't go backwards. Why? Because fundamentally there's no growing end for it to go on, right? If you're trying to go 3' to 5', what that means is you're starting from this 5' end, but what's sticking out there in their 5' end? It's a phosphate, right? You can't do anything with that. Remember, you need the elements of dehydration, which means you need water, H2O. The 3' hydroxyl is the only one that gives you that, right? That's the reason why you can only go in the 5' to 3' direction. So what this means then is this basically means the DNA polymerase requires a 3' OH to grow from. But here's the problem. If I just take this piece of DNA and I just open it up like this as a template, is there a 3' OH for this thing to grab onto? Like it has to lay down nucleotides right here. It's complementary to the template. Is there anywhere for it to grab onto to start from? Well, no, because the only 5' and 3' that you have here is associated with this strand, right? I mean, you have to have a 3' OH here somewhere to grow from to go in that direction. So you literally have nothing there, right? So the one thing DNA polymerase cannot do is start something from scratch. It doesn't have the ability to say, so there's a T. I'm just going to grab an A and then put it down there. There goes my 3' OH. It can't do that because it needs a 3' OH in order to get started. So there's a problem here, isn't there? You have the template, yes, but you don't have a place to start. The polymerase doesn't have anything to grab onto. So what do? You make a primer. So that's basically this particular piece right here. What you do is you start the process off with an enzyme that can lay down nucleotides from scratch. And this basically will make what's called an RNA primer. This is an RNA polymerase. Most RNA polymerases do have the ability to lay down nucleotides from scratch. They're different than DNA polymerases. But all a primer is just a little short stretch of sequence. That's all it is. That's complementary to your region. Like for instance, this is just 1, 2, 3, 4, 5 little pieces there that are complementary to that little stretch of DNA. And all it does is just basically make those little RNA, that little RNA stretch right there. But what it does is it creates a 3' OH. Guess what? Your polymerase now has what it needs in order to extend off of that. So your RNA primer gets laid down as a starter to give your DNA polymerase something to work from. And then once you have this, your DNA polymerase can take this and can fill in the rest. But that's an initiation thing, right? So you have to get that DNA polymerase started. It's very similar to high performance race cars, like open wheel types of situations, like Indy 500 sorts of things. Can you drive an Indy car just around town, puttering around at 25 miles an hour? No. The gearing ratio of an Indy car makes it pretty well functionless, which is at low speeds, right? Because it just has it'll bunny hop, basically is what it'll do. And so you kind of have to get it up to a certain speed. You have to sort of initiate it. You got to get it up to a place where it can sort of take over, which is also always the reason why, if you notice that when you watch races, a lot of times you'll see members of the pit crew, they'll kind of like push the car from behind, and then the car will all of a sudden kick in and take off. Why are they doing that? It's to basically get them kind of up to speed so they don't bunny hop, right? They're kind of like trying to push them along. You also see it with drag racing as well, right? A lot of times those cars, they're not just driving up to the finish line. The guys are pushing the cars up to the finish line. Why? Because they're not designed to go slow. They're designed to go fast, right? And so they're not really functional at the lower end, which is the reason why, if you think about it, the pit crew pushing the car is a lot like priming the situation, giving you the necessary speed so that when you do hit the lowest gear, which is super fast for an open wheel race car, and basically that's going to give them the ability to sort of kick in and sort of jump off of that just a little bit of speed so they can take off. And because otherwise what would happen is the car would just sit there and spin its wheels, just like you do in drag racing, right? I mean, they're sitting there, those big old fat tires. I mean, they're sitting there just spinning and smoking, and it's like takes them forever to get going. That's what would happen with an Indy car. The idea is you want to get going. And none of that, by the way, you don't want to do that to your tires. Just got a tire change, which means you're just going to have to come into the pit again more often, and you're going to be burning your tires. That's a recipe for losing the race. Similar idea, right? You kind of have to prime the pump a little bit. And that's what the primer does. So let's take a look and see what kind of lessons we learned from prokaryotic replication. Now remember, from here on out, we always learn our lessons in prokaryotes, and then we apply them to eukaryotes. So first of all, remember, prokaryotes have circular DNA. We kind of learned a little bit about this already, right? Because we kind of took a look at that in the mitosis chapter. So we saw a little bit of that. This is just kind of recapitulation of that, so I don't want to spend too much time here. We already learned about the origin of replication, didn't we? Right? In this big old circle, you have a start point. You have a place where you can actually assemble and begin your replication. So that's going to be your origin replication. You move in both directions until you come to the end, and then ultimately you come to a termination. So that's kind of what it looks like. So here's your origin. And then what's going to happen is your enzymes are going to find that area. They're going to start moving in either direction, like the rolling circles mechanism. And then they're going to terminate here at the termination sequence. And then once they get to the termination sequence, they're going to have like the connected magician's ring phase. And then those rings are going to separate to create two new genomes. And now at this particular point, you just go through cytokinesis. But you can see how you basically have one complex called the replosome on each side, basically moving in both directions. Right. By the way, sometimes you'll hear this term, replosome, spliceosome, things like that. Whenever we mention that, that's usually one thing going on, replication. That's usually there's a lot of players in there. There's a lot of proteins, there's a lot of factors. There's a lot of different protein components in there. So when there's a big complex of proteins that are involved in one basic mechanism, we usually refer to it as something like that. So replosome just is referring to this is a big old complex of lots of different proteins in there. But we're not going to talk about those. We're just going to talk about what it does as a replosome. So it's a simplification of the major complexes. So in bacteria, you have a couple of different polymerases. So in pol 1, basically, this is the guy whose job is to come in there and remove those RNA primers. Remember, we just got done priming the DNA with RNA primers. And now that the DNA has been made, we need to remove those primers, and we're going to replace them with DNA. We don't just leave RNA in there. Okay. It's the job of pol 1. We'll see pol 1 in its mechanism here in just a second. The big one that we actually studied mostly is pol 2. Pol 2 is going to be, that's in eukaryotes. Pol 2 in bacteria is basically a DNA repair. So remember we talked about DNA repair? So what this does is basically replaces damaged DNA. Now, pol 3 is the big lifter, right? This is going to be your main enzyme. This basically makes the bulk DNA. So between the three of them, you have DNA polymerase 3, which makes most of the DNA. Pol 1 will come in and remove the RNA primers and replace it with DNA. And then pol 2 specifically is only involved in DNA repair, which will actually come in and replace damaged DNA. Right, so they have three very different functions to them. Now, all of them will have basically what's called a 3' to 5' exonuclease activity. What does that mean? That basically means if you have your 5' here, your 3' here, that means it can basically chew the DNA just laid down from the reverse direction. So that's 3' to 5'. So proofread, this is like a backspace button on a computer or a reverse gear. Oftentimes, like if you're typing and you make a little bit of a typo, a lot of times before just going on and let spell check do it, sometimes you'll do what? Hit the backspace button and just retype it, right? That's exactly what this one does. So the reverse gear, the 3' to 5' exonuclease activity is your backspace button. So when you make a typo and you catch it, then it's like, oh, wait, I just screwed that up. Let me backspace, right? And then you retype it out and it's good. Okay. Paul 1 is the only one that has the forward exonuclease activity. So what does it have? So Paul 1 has both backspace and overstrike, right? Which you used to use a lot, but now you don't really use it so much anymore, right? But basically Paul 1 can chew either forward or backward, which is helpful, right? Because it has to sort of remove stuff. So it needs to be able to sort of remove things either forward or backward. So that's an important idea, right? Because even while you're making your DNA, you're already starting to correct it, right? That's what that whole backspace button thing is telling you. So you're already cleaning up some of your mistakes that you know about. Okay, let's take a look at some of these initiation enzymes. So one of the first things you have to do in order to get access to your DNA, right? You need to get access to your template so you can actually have access to the information in order to copy it. What are some of the things you have to do? Well, first of all, the first enzyme that we're going to introduce ourselves to is what's called the helicase. The helicase's job is to unwind the DNA. So I oftentimes will refer to this as the unwindase. So its job is to come in and basically unwind the DNA and to separate it. So basically, this is going to be doing both, right? You're going to unwind it and you're going to separate it out. Now, here's the problem. When you separate out your DNA and you turn it into single-stranded template, it doesn't want to stay that way, does it? It wants to basically rewind up, right? It wants to re-zipper up like Velcro, right? So what keeps it from doing that? Well, single-stranded binding proteins will basically do a couple of things. First of all, it will stabilize the single strands and protect them. So the single-stranded binding proteins will basically keep that single-stranded DNA from just like double stranding up, right? So kind of just undoing what you just did as a windase. It'll also protect it. Why does it need to protect it? What's the fate of single-stranded nucleic acid in a cell? The fate of all single-stranded nucleic acid in a cell is to get degraded. So if you've got single-stranded nucleic acid in a cell, those exonucleases will go after it and we'll start chopping it all up. And this is something that actually in eukaryotes in particular, not so much in prokaryotes, certainly, but in eukaryotes, definitely. So for us to have our DNA in a single-stranded state is very dangerous. Why? Why do we degrade it? Well, the reason why we degrade it is because by and large, viral genomes are single-stranded. So usually if we see single-stranded nucleic acid in our cell, we think viral genome, kill, right? So the idea is if you kill off and digest up the viral genome, then you will not get virally infected, okay? So if you're gonna have single-stranded nucleic acid, you have to have some sort of a strategy to protect it. And there's lots of strategies to protect it. One of them is with single-stranded binding protein. So it does a couple of things, right? It keeps that single-stranded template open and exposed, and it also protects it. The other big one, the other third one is what's called topoisomerase, of which gyrase is a type of it. So what this does is it basically prevents supercoiling, which you can kind of see here. So supercoiling basically is torsional strain. In the DNA molecule. So think about it. It's very much like a rubber band, right? If you were to take a rubber band and roll it between your fingers, what happens to that rubber band? Yeah, it gets all twisted, right? And it kind of forms little loops. And if you really get in there, I mean, really kind of work it, it's going to start to fold up on itself, right? It's going to create all this. So what you're doing is you're creating torsional strain in that individual because of all the twisting. Now, how do you relieve that strain? What's the easiest way to relieve it? What? Well, that's true, if you let it go. But what if you didn't let it go? You couldn't. Yeah, just cut one of the loops, right? As soon as you cut a loop, what happens? All that torsional strain gets released. That's exactly what topoisomerase is doing. So topoisomerase basically is moving ahead, and what it does is it basically nicks the DNA. To relieve that torsional strain. Not torsional stain, torsional strain. Right? So that's what it does. So it kind of goes forward ahead of the replosome and is kind of smoothing it all out. And as it does that, it's going to be like nicking the DNA and releasing that torsional strain so it can kind of smooth it all out. That torsional strain is building up because you're untwisting and twisting and things like that. Okay, so that's building up in your system. And so you kind of just nick it to kind of smooth it all out. So that one kind of heads back. So those are three major enzymes associated. We've actually already been introduced to four major enzymes actually in the replication process, right? Actually 5. We've seen Pol 3, which is the heavy lifter, right? We've seen Pol 1, which is the replacer. Now we've seen helicase, which is the unwindase. We've seen single-stranded binding proteins, which are the stabilizers. And now we've seen topoisomerase, right? Which is the strain reducer. So we've already seen 5 enzymes involved in replication. So now what we want to do is take a look at the mechanism of replication. And I want to kind of get this introduced first, and then we'll take a look at the details later. But one of the things that we noticed with replication is that it is semi-discontinuous. Now, there's a whole thing with the models. Oftentimes, it's always trip students out there is continuous and semi-discontinuous and things of that nature. But basically, what semi-discontinuous essentially means is that the 2 strands don't have the same mechanism. Okay, so discontinuous means that if you get started, you just go until you're done. That's continuous, right? Discontinuous is that you're kind of like jumping around. So you kind of do one batch, do another stretch, and then it's kind of like a disjointed kind of a process. When you're semi-discontinuous, that means a part of you just gets started and does it from the beginning. So that would be the leading strand, which is continuous. So once you start, you just continuously make it from that initial primer until you're finished. So that's a continuous process. The other strand is what we call the lagging strand, though. The lagging strand essentially is a discontinuous thing. It basically makes chunks. Chunks of DNA, and then it stitches them together. So those fragments of DNA are what we refer to as Okazaki fragments. So what we noticed was that the reason why we noticed this was because it's a very easy thing to do. Take a dividing piece of DNA and then anneal it, that is heat it up so it falls apart, and then run it out on a gel. If it's completely continuous, then you would only see one band size associated with both template, right? They'd be the same size. But what you saw was one band that was heavy, consistent with the complete template, all these little fragments, right? Little different sizes. And those are called the Okazaki fragments. So you notice then that there was something else going on here. And so we actually have two different ways or two different mechanisms basically of making DNA. So when we have essentially our DNA, we have to unwind it and we have to open it all up. And so you can see, first of all, on one side, you're going to basically make a primer, right? That's going to give you your 3' OH. And that's going to allow your polymerase to land on there and basically just go, right? And then you continue to open up what we call the replication fork. And it continues to open up. But the continuous side just basically goes right on in there and just kind of keeps plowing on in until it's done. All right, never stops, doesn't have to stop. On the other side, however, once you prime one event and you basically fill in the gap, guess what? All the new open stuff is behind you. So that means you have to have a way of going behind you and doing the same thing, lay down a primer and then fill in the gap. So this would be the leading strand, the continuous strand. This would be the lagging strand. This is the way it looks. This is the way I like to think of it, right? Now follow me here. I'm going to draw a line. You're not going to be able to tell the difference if you see it from the beginning versus the end. You have to actually watch the process. I'm going to draw a line that's consistent with both of these leading and lagging strands. The first one is a continuous. That's the leading strand, right? So in the leading strand, it's like if I were to start from here to here, connect those dots. The leading strand would basically start from this end and then basically continuously move forward until I hit my destination and I stop, right? That would be continuous or leading. Now, what is discontinuous? This would be the lagging strand scenario. Now notice, I'm going to have the same destination. Only in this case, what's going to happen is I can only go forward. I can't go backward. So here's what's going to happen. I basically am going to draw the line like this. I'm going to start here and I'm going to connect. And then I'm going to fall off and I'm going to start and put my pen down here. And then I'm going to add that piece. I'm going to pull my pen off and I'm going to drop it right down here. I'm going to connect that piece. I'm going to drop it off. I'm going to put my pen right there. I'm going to connect that piece. Different process, yes? So that's exactly what happens between leading strand and lagging strand. So the question is, what does that look like in terms of our DNA and the enzymes we have? Well, in both cases, we start off with our helicase, right? So that's our unwindase. So this basically opens up our replication fork, right? That's this guy right here. That exposes our template. So there's our replication fork. Now the next step, right? This is initiation, by the way. This is part of initiation. The next step is to prime it, right? You got to lay down primers. So who does that? Well, let's add our sixth enzyme. So we actually have an RNA polymerase called DNA primase. And this one basically will make the RNA primer like this. There it is. Just that easy. Lay down a primer right there, and it'll lay down a primer right here. And it basically provides those 3' OHs. So now your DNA polymerase can land right here and start moving in. It can land right here and start moving out. So your primase is the sixth enzyme that we've been introduced to now, whose job it is to lay down these RNA primers. Okay. So we're at about our witching hour. So it's a good place to stop, and we'll talk a little bit about the mechanism, the pol 3, and we'll kind of get into the eukaryotic scenario. So this is a good stopping point for us. We should be able to finish chapter 14 pretty easily on Wednesday, and then get a healthy start on 15. So